Modern diagnostic applications call for accurate and dynamic sensing of key analytes in biological samples. Nanosensors, an emerging field in diagnostics, possess many advantageous properties for spatial and temporal sensing. These small particles possess an inert polymeric shell which encapsulate various sensing components within the core. The inert shell minimizes the invasiveness of the particle while allowing small molecule analytes to diffuse through the polymer matrix to the analyte-sensitive component and can be detected based on their optical properties. These sensing components are modular and allow us to customize our sensors to fit the given application. Oxygen is a very commonly studied analyte because of its relation to metabolic activities in aerobic organisms. Nanosensors can be made which possess fluorescent dyes that are quenched in the presence of oxygen, and this change in signal can be used to measure the concentration of oxygen in the sample. In bacterial biofilms, oxygen nanosensors can tell us much about the behavior of these cells by sensing dynamic changes in the biofilm's metabolic growth over time. Other sensors are typically more invasive and cannot capture spatial oxygen gradients, therefore, nanosensors could give us more useful information. While there are a variety of dyes that are sensitive to oxygen as well, we use a dye called platinum octaethyl porphyrin ketone, or PTOEPK, because of its unique spectral properties to emit in the near-infrared wavelength range. This is an important characteristic of the nanosensor because in many biological samples, background signal can be very high at visible wavelengths. We pair this oxygen-sensitive dye with a constant reference dye, dye D, which can be used to normalize the signal intensity to the concentration of nanosensors and the optical properties of the sample environment. These dyes can be excited at the same wavelength, but can be differentiated through an emission spectra captured at different wavelengths. In order to calibrate these sensors, we use a gas bubbling setup to control oxygen concentrations in the sample containing the nanosensors. At atmospheric equilibrium, the sample will have a maximum concentration of 21% oxygen dissolved. Nitrogen gas can be used to exchange dissolved oxygen from the solution, allowing us to lower the oxygen concentration to 0%. By adjusting the gas flow mixture with a controlled ratio of air and nitrogen, we can generate any oxygen concentration between those 2 points and measure the fluorescent signal of the dye. To generate a standard curve, we use the Stern-Volmer equation, which relates the fluorescent signal in the absence of the quenching component and the signal at various concentrations of the quencher as seen in the diagram. We can then determine the Stern-Volmer constant, which allows us to determine the oxygen concentration of an unknown sample. Oxygen nanosensors have previously been demonstrated to be effective at sensing oxygen in biofilms in vitro using the dye PT-TFPP, which emits in the visible wavelength. However, we would like to translate this work to a near IR dye, such as PTOEPK. When biofilms are active, the cells rapidly take up oxygen from the surrounding biofilm matrix. However, if we treat those cells with high concentrations of antibiotic, we can detect the change in activity as oxygen will be restored to the biofilm. The figure at the bottom right shows images taken by a confocal laser scanning microscope of a Pseudomonas aeruginosa biofilm after being treated with colistin sulfate, a commonly used antimicrobial agent. The green signal represents the oxygen-sensitive dye, while the blue signal represents the reference dye to show that oxygen concentrations in the biofilm increased as the antibiotic takes effect on the cells. Many biofilm infections, however, possess higher antibiotic resistance compared to planktonic bacterial strains. In order to find the most effective treatment plan for infected patients, we pose using oxygen nanosensors to determine the type of antibiotic and the antibiotic concentration which is most effective in eradicating the infection. Additionally, nanosensor components can be swapped for new analyte sensing dyes, allowing us to extend these applications to anaerobic organisms which metabolize sulfur or iron instead of oxygen. Finally, the near-IR probe may allow us to extend this application to in vivo samples through the imaging of live tissue cells, enabling us to monitor the infection in real time.